Thanks for having me here. It's really good to be back to Vancouver. I was here actually in October for the ASHG, and I think I see some familiar faces here. So, so today I want to tell you about the 10x single-cell platform and some of the applications we have on the brain and immune cells. So to start off, I just want to acknowledge everyone at 10x Genomics who made this awesome products, the genome as well as the single-cell products. I also want to thank my collaborators on one of the studies that we'll talk about today, which is transplant samples from AML patients. So, so the reason we're interested in single-cell RNA sequencing is because cell is a fundamental unit of biology. So to really, like in mammals, every cell starts with a zygote, which then develops into a blastocyst and then into all three germ layers. So to really start understanding, the system as a whole, we need to understand sort of the individual components that make up the system, and single-cell RNA sequencing give us a tool to do that. But the existing tools for single-cell RNA sequencing analysis are limited from several perspectives. First is, a lot of the tools only allow you to look at a few number of cells. And some of the tools out there, you can look at more cells, but then you have to do enrichment and sorting cells into plates, which becomes very cumbersome and difficult to scale. And some other droplet-based platforms requires a custom setup of microfluidics pumps. So that's, unless you are an expert in microfluidics, sometimes it's a little bit tricky to get that going. So to overcome those challenges, 10x have come up with a microfluidic system that is high throughput scalable and does not require a custom setup. At the core of the technology is a nanoliter-sized droplet that encapsulates a single cell and a single gel bead. The gel bead consists of oligos that are necessary to index cells and index transcripts, as well as primers that are needed for the reverse transcription. So, and our platform can accommodate 8 samples at a time, and you can process cells up to tens of thousands or even millions of cells at a time, or even millions of cells. So the cell capture rate is highly efficient, and the doublet rate is low. So which makes a lot of the applications feasible. So just to illustrate what we mean by doublet rate, so we perform sort of a classic experiment in single-cell RNA sequencing in which we combine mouse and human cells at roughly equal ratios, and we're looking for droplets that contain a combination of mouse and human reads. So those droplets are indicators of a doublet, because if it's a purely single cell, you should just detect mouse or human, but not both. And based on this method, we were able to estimate our doublet rate is around 0.9% for every 1,000 cells, and it scales pretty much linearly with the number of cells that we use. So this is demonstrated here on this plot. We tested on 4 different cell loads, so ranging from 100 cells recovered to over 13,000 cells recovered for a sequencing run, for a library run. And so you can see the doublet increases from 0% to about 10%. And another thing I want to point out is the efficient cell capture rate. So, for example, if I want to, end up with about 1,000 cells in my library, I only really need 1,500 cells to begin with. So this is how we calculated about 65% cell capture efficiency. So this is orders of magnitude higher than a lot of the competing single-cell platforms out there. And this allows you to, start looking at samples in which the input, is quite limited. So frankly, a lot of the applications that were not previously possible can now be done on this platform. So, so we actually released our single-cell product at the last year, and at the end of this last year, we released sort of a version 2.0 of the product. And the version 2.0, it's much more streamlined, and it's a lot more sensitive. And there are multiple ways for you to look at sensitivity. But one way to look at it is to look at the unique number of molecules or unique number of genes that you can detect in each cell. So in this case, I basically, I'm showing the plots for the two well-known cell types, the 293T human embryonic kidney cells and the 3T3 fibroblast in mouse. So, and when we sequence those libraries to saturation, we're able to detect about 8,000 genes so unique genes per cell. And then if you look at the transcript level, we're looking at about 20, I think it's about 80,000 unique transcripts per cell. So that's one way of looking at sensitivity. And so since then, we've demonstrated a variety of applications in multiple systems encompassing all the genome layers. So today, I actually want to talk about some of the data we've collected in the brain, in mouse brain. So that consists of 1.3 million cells. And I also want to tell you a little bit about some analysis we have done on the AML patients. And what's also really exciting is some of our collaborations with different academic groups have started to get published. So I want to point out one of the publications that's coming out in Nature in a couple months is looking at mouse intestinal epithelial cells. So we profiled about 10,000 cells from mouse intestine, and we're able to look at drivers and that affect the pluripotency and proliferation of those stem cells. So I encourage you to take a look at the publication when that comes out. So before I move on, I'm trying to go quickly so I can get you guys out here in time. But before I move on to sort of start looking at the brain and the immune cells, are there any questions about just the system in general? Okay. So yeah, so for the brain cells, so we started thinking about this project when we are releasing when we're introducing our version 2.0, and we were thinking, what's a good way to demonstrate an experiment that nobody has done before, and that uniquely showcase the advantages of 10x genomics, but also providing some, interesting scientific data to the community. So we came up with, a million cell experiment. To do this, we purchased. So we don't really know how to work with mouse brain, so we just purchased a mouse brain from a commercial vendor. And you can specify, the regions that you're interested in. So we bought cortex, hippocampus, and ventricular zone from embryonic mice, and so they dissected everything for us and sent us the brains on ice, and then overnight, and we put them in 4 degrees until we're ready to make the single cell libraries. So on the day when we're making them, so we just dissociate the brain. And used about 17 microfluidics chips to make 133 single-cell libraries. And that all happened over a course of 2 days. So with 3 people in the lab. So then we sequenced all of the data on 11 high-seq 4000 flow cells. And at the end of the day, we're able to retrieve about 1.3 million cells with a sequencing coverage about 18,000 reads per cell. So, and just want to put this experiment in perspective. So we look at all of the single cell experiments that have been done in the nervous system, and plotted them, the number of cells profiled cumulatively over time. So the first jump you can see here, this 10x jump comes with the introduction of the academic droplet system. And the second jump here is the introduction of our million-scale brain cell experiment. So it is one thing to actually to collect all of those libraries. It's another thing actually to even process the data. Because we got 3.6 terabytes of ASCII files. And so we needed to figure out a way to process those in time with the amount of memories and CPU powers we have. So just to give you a sense, first we need to process every single library, and that's about 200 CPU core hours. And then so that times 133. And then after we process each of the libraries, then we have to aggregate them. Because we know, we want to look at a million cells all together. We don't want to look at them separately. So to be able to aggregate them, that's another 700 CPU hours. And then after you aggregate all the data, we need to do some sort of dimension reduction and clustering to start understand what the data means. That's another 350 CPU hours. So altogether, it's about 1,200 CPU hours. So so that's like over 50 days of just computing, just data crunching, and not mentioning the large amount of memories that are necessary for this. And there's an extensive engineering and re-optimization for a lot of this process to even make it run, with 300 gigs of memory. So. But at the end of the day, this is what the 1.3 million cells look like. So this is what you're looking at is a 3D T-SNE plot. So what T-SNE plot is every dot here represents a cell. So there are about 1.3 million dots here. So the spatial clustering of the dots indicates sort of how closely they are transcriptionally. So cells that are similar in function or in identity will probably form clusters around themselves, and then the cells that are further apart will be further away on this plot. So and here we've color-coded the points to indicate their inferred cell identity. So since it's actually not easy to manipulate 1.3 million cells. So I'm actually just going to, I will start by showing you some of the analysis we've done looking at a randomly sampled 20,000 cells. So this is a tiny fraction of the data we collected, but we believe it's a representative snapshot of this data. So first we wanted to see if the cells are healthy. What is sort of the gene distribution and transcript distribution. So, overall, it's pretty uniform, and we saw very minimal signals indicative of cell death. And then we've basically, color the cells based on some of the well-known markers. So, for example, we can look at the Stathman-2, which is a neuronal marker. So that highlights about 75% of the population. And then you can also zoom in to look at the subsets of the neuronal cells. So there are excitatory cells and there are inhibitory cells. And then you can also look at the non-neuronal cells, which consists about 25% of the population. So one of the markers we looked at is HES1 that highlights the glial cells. And then among the glial cells, there are different subtypes like the astrocytes and oligodendrocytes. So this is just looking at a few markers. But if we look at overall all of the clusters, we were able to identify over 20 clusters. And And then here I basically color-coded them and sort of try to indicate the type of the cell types that we think they are. So so you can see that even though we've identified 8 major cell types, there is extensive structure even within each of the main cell types. So another question is, we've collected all this data. How relevant are those transcriptome profiles? Are they sort of similar to what other people have collected? We wanted to sort of get a confirmation. So one way to do that is sort of to compare that to published data. And what I'm showing here is our comparison to this published data set from Allen Institute for Brain Science. So they've collected about 1,700 cells from different parts of the brain. And they actually have to do extensive enrichment, or in some cases use transgenic mice to collect some of the really rare cell types. So, but then so we were able to compare our single cell data to theirs just by doing a simple correlation test to see how correlated is our single cell profile to theirs at every single cell level, and we color coded them based on the best match to their reference data set. So overall, you can see the cell types that were inferred from our marker-based study is actually highly consistent with our comparison to this external data set. And we also sort of did the comparison to other publicly available data sets as well, and they're all very consistent. So another thing, one more feature I want to point out is that those, within, I alluded to this earlier, within each main type, you can see a lot of substructure. So then we can zoom in, to those substructure and then further identify novel cell types. So in this case, I basically zoom into the inhibitory neurons, and that constitutes about 15% of the population. And we're able to identify, another 12 clusters. And then you can compare those to, again, this reference from the Allen Brain Institute, and start to notice some of the really rare inhibitory neurons. So that they have actually used lots of mice and transgenic mice to collect those kind of data. But with 1.3 million cells, you don't really have to do that. That makes the that makes the sample prep and the experimental design much easier. But you do have to be able to analyze the data and be able to tease out the different cell types. So we have made all of this data publicly available on our website. So you can download all of them from the FASTQ files, as well as the alignment and processed matrix files. Actually, quite a few academic groups have started playing with this data set, and we already saw one publication in bioRxiv analyzing our data. And I recently went to a couple of conferences in which people presented analysis of our data. So, so this is already sort of inspiring people to develop methods and to also encourage the neurobiologists to really start identifying novel cell types. So, and So, and since the release of the data, we've also had a lot of interest from people just wanting to download this data set, but also a lot of people asking us, after I download the data, what do I do with it? Because they can't really, open up those the data in R. So, there isn't enough memory, and also R uses this 32-bit encoding for integers, which is not enough for this data set. So, we've actually put out some tutorials on how to even start looking at the data. But also today I want to take this opportunity to introduce you to our LoupeCell browser, so which is sort of an interactive tool we recently released about a month ago. So you can actually open up that 1.3 million cell data in this cell browser and start playing around, and then start making discoveries. So I have the web browser on my laptop, and then also this data on my laptop. So I was going to give a live demonstration, but because we changed the laptop, so I'll just show you some of the screenshots, and then you're welcome to download this data online. And this browser is also freely available on our website. So anyway, so when you open it up, it looks like this. And then you can basically just click, drag or browse, like a LoupeCell browser file from your desktop, and then, be able to open it up. And this is what 1.3 million brain cells look like in 2D in the LoupeCell browser. I think it actually kind of looks like brain to me, although I'm not really a neurobiologist. And then, so on the left of this browser, you can see all of those different ways to manipulate. We kind of sort of structured it like Illustrator kind of thing. So you have all of those tools that you can manipulate, and you can zoom in. You can zoom into just one cell, or you can zoom out to look at all the 1.3 million cells at a time. And on the right here, there are some of the control panels. So one of them is showing all of the clusters that we identified. In this case, I think it's over 50 clusters in this 1.3 million cells. And then you can also, you may want to just look at the expression of your favorite gene. So you can just type it here under the gene expression control panel, and then it starts to highlight cells with different expression level of your favorite gene. So in this case, we're looking at Staffman2, which is the neuronal marker. So this is highlighting about 75% of the population. And you can also look at the markers for inhibitory neurons and excitatory neurons. And this is a marker for the glial cells. And in addition, you can also do differential gene expression analysis between different clusters. So, and then that information is all captured here in this heat panel, in this heat map. So, so then, for example, if I click on, cluster 7, I can start to see some of the markers, that are enriched in cluster 7, and then you can actually then say, okay, now I want to plot it to see how it looks like in this, on this T-sleep plot, and you can, see some of the so cluster 7 obviously is highly enriched with this expression of this marker, but you can also see this markers in some other cells. So all of those are inhibitory neurons, so that makes sense. So anyways, I think hopefully this will be a good tool to help people dive into the biology, and you can do all of those without having to write a single line of code. And all of those can images and some of the analysis, differential gene expression analysis can be exported as well. Okay, so this is the section about brain cells. So are there any questions before I move on? Yeah. This is 3 regions mixed. So the cortex, hippocampus, and ventricular zone. So there are actually, I didn't go into it here, but you can actually, there's enough information there for you to see different parts of the cortex and hippocampus, like different layers, if you start digging into the different clusters. And then here, I'm showing you basically the data from 2 mice. So you can actually see which one comes from which mouse. Yeah. So one is a female and one is male, and you can actually see which one is which. So there are some signature genes Yeah. Yeah. Well, that was our intention, but we couldn't find someone who can give us the brain. So we contacted a few neurobiology labs at Stanford, but they couldn't really give us the material in time for us to do this. But this is the embryonic time point. We actually do have an adult sort of P6 time point. We collected all of those data, and we sequenced some of it, but we haven't sequenced. We didn't sequence 1.3 million cells. So that data is available, and when we did some comparisons, we definitely noticed the different proportions of neuronal classes. But as of now, we don't really have any plans to publish any of this or to do more. But if there are people here in the audience who are interested, we can consider sharing some of that data. Yeah. I think maybe Eric may be a better person, Eric, to answer. It's 133 libraries times 1,200, right? 11. Yeah, yeah. But the other thing to consider is, when Allen Brain Institute did it, they had to carry out all those transgenic mice, for a certain period of time, and they have to, I don't remember exactly how many embryos they used, but in some. And in some cases, in some other studies, they used hundreds to thousands of embryos to be able to collect a few hundred, single cell. Oh, is this better?
Yeah, sorry about that. I was saving your eyes.
So, should we go back to?
Yeah, maybe you want to.
Should we go back to some of the? All right. Oh, maybe we can watch the 3D video again. Yeah. So so some studies, in order to collect 1,000 embryos, they had to basically sacrifice tens of thousands of embryos, right? For mice, it's maybe relatively easy to do, but still, tens of thousands embryos it's a lot of work, and also probably costs a lot of money as well. So you can think about it this way. Okay, so yeah. In total, yeah. Yeah. I don't remember how many instruments we used, but we made 133 libraries in 2 days. Yeah, by 3 people. So first day is primarily just getting single cell suspension. Yeah. So each run, so we used the 17 chips, I think. Each chip, to run each chip only takes 7 minutes, right? So, so that's not a ton of time. So a few hours you can process all of them. Yeah.
So that seems like a really great resource for this is what could look like in the future. But is there a lot of variation within sites? Like how good is the representation? We're thinking of this amazing million cells.
Mm-hmm.
But can we use that as a control, like as a reference for
I think, yeah, so there's definitely variation, and the fact that we can see two, sort of mice with similar genetic background to have different clusters already show you, demonstrate, the variations within this single cell data. But there are ways for you to remove certain variations, and you can sort of only focus on the signal. So we haven't done much of the pre-processing here, and I think a lot of the people looking at this data are trying to do some of those themselves. And this also comes back to this question. 10x is sort of intimately involved in this human cell atlas project, in which they want to sequence of 1 trillion cells in human. So, one of the things we have been talking to them is how do you have some sort of QC standards, and how do you compare across all of those different methods and different sequencing centers? All of those, it's not possible for one center to sequence all 1 trillion cells. Yeah, so that's an ongoing problem, I think. Yeah. Okay, so I missed the first part. Is there a way to? Oh, you mean have like an external marker? Yeah, yeah. I mean, I think that's sort of how the basically some of the experiments are done with the Allen brain Alice thing. So they were sorting cells based on the fluorescent markers, and then so they have some sort of a comparison. But we wanted to sort of come up with a way that removes any of those sort of pre-processing and sample prep. So we don't have that. But you can definitely do it first and then run it on our system. It should still be compatible. And we've done that with some of the immune cells. Yeah. Yeah. We have seen some indications, but because we're not really mostly I don't have a neuroscience background, and so I haven't really spent the time to really dig into the data, and we kind of just thought, we're just going to collect this, put this data out there for people with expertise to really look at it. And we have some people, neuroscientists, contacting us about some of the things that they're noticing, and we're trying to support them to publish their findings. We're not going to try to publish anything, and also the fact is because we don't really have a good way to manipulate, different parts of the brain and also manipulate cells. So we would have to collaborate with someone anyway to validate our findings. Yeah. Black 6. Yeah, it was when I was trying to set up this experiment, I contacted lots of companies asking if they can provide us something slightly more interesting samples and or some different mice with different genetic background, but it was not easy because unless you set up a collaboration, you don't have easy access to those samples. Yeah. So, but hopefully this is just a starting point to inspire other people to look at, to think about, to design and think about a million-cell experiment that they can look at. Now, of course, with the Human Cell Atlas project, this is something that people have been thinking a lot about. So, and they're definitely going to include the healthy as well as diseased samples. Okay, so we're running short on time, so I'm going to actually just make this second section really fast. So this is actually published work. It came out a couple of months ago in Nature Communications, and we wanted to sort of look at immune cells and in the context of AML, oftentimes. So those samples are coming from transplant patients. They have AML, and I'm going to skip that introduction. And so those transplant patients, they have AML, and the standard therapy is they go through sort of a radiation or chemo. But then when those fail, then they undergo this hematopoietic stem cell transplant. So it's basically a bone marrow transplant. So you get it from a healthy donor, and then you put it into a host. And then the doctors have to monitor the progress of the host over time. So during this time period, they need to monitor sort of the proportion of the donor versus host population. They need to know sort of the type of the cells that are there. But then they need to look for any kind of a signs of a relapse, if there's any rejection as well. So there are different, clinical tools, assays that allow you to answer those questions, but they're not. But you have to use different tools for each of those questions. So first, people, would do a FACS sort to isolate different cell types from the bone marrows, and then for each of the cell types they isolate, then they do some sort of a PCR-based test. Look at some of the short tandem repeat markers. So this test becomes really tedious, if you want to look at 10 different cell types, and then you have to do the PCR assay for all of the 10 cell types and compare that from donor to host, right? So and also at the end of the day, you're still getting really bulk information. There's no way for you to look at every single cell to tell if it's a host or donor and the cell type it represents. This kind of questions are really relatively easy to address with single-cell analysis because with the RNA-seq data, we already know which cell types they're coming from, based on our clustering, and I demonstrated that earlier with the brain population. But you can do that with the bone marrow cells, so you can look at the T cells, B cells, monocytes, and all of those. And then in addition, because we have sequencing reads for all of those cells, so we can look at all of the reads from every cell and then start to identify variants. So some of the variants will be shared among the donor and the host, but some variants would just be unique to the host or the donor. So we could use those variants to start, distinguishing the donor-derived cells versus the host-derived cells. So we build a probabilistic model to look at our variant data and then basically say, what's the probability that the cell comes from a host and what's the probability that the cell comes from a donor. And we do this all with single-cell RNA-seq data, not with any of the pre-existing knowledge of the variants. So after we built the model, we wanted to validate our model on to see how good the model is. So one controlled way to test this is we have two populations in which we know fairly well, and then we mix them at different ratios. And then we ask if our model can predict the correct ratios, right? And then the correct identity. So this comes down to looking at the sensitivity and PPV. So our models. So looking at doing this analysis, our model shows about greater than 95% accuracy. When the minor population is more than 3%. So when the minor population is less than 3%, our model is not as sensitive. But so applying this model to some real patient data, so we were able to get archived bone marrow mononuclear cells from 2 AML patients. So before they received transplant and after they received the transplant. And then we also collected some cells from healthy controls. So altogether, we're looking at 17,000 cells, and this is what the RNA sequencing profile looks like on the t-SNE plot. So this is about 17,000 points here, and then you can start looking at the markers and then start identifying the subpopulations. So we're able to identify a variety of the cell types. So one example is you're looking at the red blood cells, and you can see really immature red blood cells that are the blast in nature, and then all the way to mature erythrocytes. And there are also different immature granulocytes and monocytes and so on. So next, then we looked at the individual samples. So this is one of the leukemia patients before the transplant. And you can see, compared to the healthy control, there's an expansion of the monocytes. And this is consistent with the clinical diagnosis of this patient. This person has monocytic leukemia. So, and then if you look at the post-transplant sample, our variant analysis indicated that there's only one basically species, and it's all of this 100% donor-derived. And again, this is consistent with the clinical diagnosis of this patient, that this patient is cured after the bone marrow transplant. But with the second patient, this is very different. So first, you see an expansion of the red blood cells. And then this is consistent, again with the clinical diagnosis. This person has erythroid leukemia. And then if you look at the post-transplant sample, and now we're looking at 14% of the donor cells and 86% of the host cells. And then you can sort of, so this is one immediate indication that this person had a relapse, right? Because the host cells are taking over the donor cells, and then basically outgrowing the healthy donor cells. So, and this person actually and this is completely consistent with the diagnosis of this patient. And then you can compare the host cells after the transplant to before the transplant, and start to understand some of the molecular mechanisms behind it. So, for example, you see an expansion of this blast cells in the host that wasn't as prevalent before the transplant. You can also see an expansion of the immature granulocytes. All of those are potential clues to what was going on with this AML patient. So our collaborators are expanding this study to look at more patient samples and to look at that are undergoing transplants, and trying to potentially come up with a clinical diagnosis or clinical tool to diagnose those patients better and earlier. So, but then just lastly, I want to tell you, so again, you can use the LoupeCell browser to start playing with this data set. And we actually have a tutorial on our website if you follow this link to start looking at different patients and different cell types, and then start looking at the differential expression analysis. So in the last minute or so, I just want to tell you what is coming up from 10x Genomics. So we released this single-cell 3' assay to look at the RNA sequencing, but we're also coming up with a single-cell 5' assay. So the advantage of 5' assays, then you are looking at transcription start site. So this allows you to look at a lot of things that are not possible with when you look at transcripts from the 3' end. One example of that is to look at the T-cell receptors and B-cell receptors. And so why is that interesting? So T-cell receptors and B-cell receptors are the workhorse of the adaptive immune system. So each cell, if you look at T-cells, has T-cell receptor alpha and T-cell receptor beta, and the unique combination of alpha and beta can confer a specificity to a specific antigen. And same goes with the B-cells. So knowing the combination of the alpha and beta, or the combination of heavy and light chain, can help the clinicians and pharmaceutical companies to develop cancer therapies. So immunocancer therapy is extremely important right now. So this is going to become an important tool to help guide people to design chimeric T cells and to start figuring out what antigens are the most specific to your responding to your immune system, and then this also has important applications in vaccines and as well as the allergy treatment. So here's just a sneak preview of some of the data that will come out. So we have profiled some of the clonal T cells. So this is from a T cell line, and you can see that every cell in this line pretty much have one type of T cell receptor. And this is very different if you take a healthy person's blood and then look at the T cells receptors from that person's blood, because every cell pretty much has a different T cell receptor. So we're looking at over 4,000 cells here, and we detected over 4,000 clonal types. So what I haven't shown you here is now if you take a patient, someone with some sort of a leukemia, where you're looking at sort of a tumor-infiltrating lymphocytes, this picture looks very different because you start to see expanded clonal types within this patient, and those are the the cells that you want to really understand and then you want to target. So that's coming out. That's going to be released in a month, and I think that's everything that I have here. So if there's any questions, feel free to. Yes. So in this product that we envisioned, we can look at the T cell receptors as well as some of the phenotype markers. So then we can sort of identify the type of T cells. So whether it's T helper cells or memory cells, naive T cells, you can look at. All right.